at noon, and this is the ACMT Grand Rounds series, and I have a distinct pleasure of introducing Dr. Michael Twiner today. Um, just some quick housekeeping. Please, we're going to try to save our questions to the end, but if there's some burning question, feel free to send me a message in the text function or raise your hand. I'll be monitoring to see if anybody has any burning questions, and if it's time sensitive, we can try to interrupt, but otherwise, for the most part, we're going to try to save our questions to the end. Once you do get a question answered, please drop your hand because I don't have the capability of lowering your hand in the raise hand function. And as always, please, if you see any great lectures or you have any specific desired topics, either send an email to myself or the ACMT office, and we can look to include those in future webinars. So Dr. Twiner is an emergency physician at Detroit Receiving Hospital and Harper University in Detroit, Michigan. But he has a previous career, a 20-year career, as a molecular toxicologist with a focus on algal toxins. He spent several years working at the National Oceanic and Atmospheric Administration Center for Coastal Environmental Health and Biomolecular Research in Charleston before developing his own research laboratory at University of Michigan. And as I understand it, he's now branched out from algal toxins to include some other realms of inquiry. But today he's going to talk to us about algal toxins, toxidromes, molecular mechanisms, and human health effects. I'm very excited. I think it's going to be particularly high yield as far as medical toxicology boards. So hope the fellows are all paying attention. And with that, I'll turn it over to Dr. Twiner.
Well, I appreciate it. Thank you very much. It's a nice introduction, and I'm happy to be here. And I really do appreciate the invitation from ACMT as well as Nick helping host. So my topic today, as Nick has already stated, has been algal toxins, and I'm going to be reviewing some algal toxins as they pertain not only to North America, but also some flavor of global health as well. And so hopefully this is somewhat relevant to the audience here. The objectives of my talk is really just review some of the more commonly encountered toxins. This isn't an all-encompassing list, and specifically those that are more of a threat to human health. Understand those general toxidromes that are associated with the specific toxin groups, and understand that they are geographical variants associated with them. I hope you also come away with a basic understanding of the mechanisms by which these toxins cause those adverse health effects. This is just some general pictures of more or less accumulated algae, and that's, algae are what producing these algal toxins. And I do want to put this in perspective that really 99% of algae are good. And I think the same can be said about terrestrial situations when it comes to plants and trees, where only 1% of those maybe plants or trees might be producing some sort of secondary metabolite that might have an adverse effect. Well, the same can be said about aquatic systems. There's really about 1% of algal species that might be considered to be harmful and produce some sort of secondary metabolite that is of relevance to us in terms of the toxicological side of things. These particular toxins are produced by a vast array of different species, and they generally fall within three different categories, both eukaryotes as well as prokaryotes. The dinoflagellates, which are eukaryotic type of species, as well as diatoms, also eukaryotic, can produce several different toxins, and we're going to go through many of these: saxitoxins, brevetoxins, oxalic acid, ciguatoxins, azospiracid, and yesotoxins. Then there's the diatoms, which are well known to produce a single particular type of algal secondary metabolite, domoic acid. And then there's the prokaryotes. These are cyanobacteria. A lot of people refer to these maybe as pond scum, and they can produce some secondary metabolites that include saxitoxins, microcystins, and nodularins. Now, harmful algal blooms can be the source of these potential toxins. And like I said, these are global issues, not just North American issues. And you can see here, many coastlines around the world have known toxins with known human health side effects and poisoning events. More specifically, here in North America, you can see there's really not a coastline that's not affected by some of these algal toxins. And just by the pictures of the algae as well as the toxin structures, you can see there's great diversity in terms of those different types of species and toxins. And that also relates to great diversity in the mechanisms by which they produce their toxicological action. This is more of a close-up version of some of those toxin structures. On the right, you see more of the lipophilic toxins, brevetoxin, yasotoxin, azospiracid, ciguatoxin, oxalic acid. And on the left, you see more of your lipophilic, hydrophilic toxin structures. These are the saxitoxins, domoic acid, microcystin. And these can be highly toxic. And this is just a general list of LD50s based on mouse in vivo studies where they're intraperitoneally injected. And you can see, orders of magnitude differences in terms of the LD50s associated with these different toxin groups. And these are the ones that I'm going to go through today because I think they're most relevant to human health effects, both in North American as well as more global waters, and that I think would be more relevant to the audience here today. Now, this is just a list that was put together of both synthetic as well as not natural compounds. And you can see here, this is the top 25 list from the most toxic at the top, botulism toxin, all the way down to the 25th. And the one thing that I wanted to point out here is that 7 out of the top 25 listed are produced by algae. Just some general kind of food for thought in terms of how these toxins do affect humans. And part of my previous career was not just affecting human health side effects, but also at various levels of the food chain and food web, including at the lowest level when talking about fish and crustaceans, but also marine mammals, birds, and then ultimately, I think relevant to this audience are human health side effects. So we'll start with ciguatoxin. This happens to be the most toxic one that we're going to talk about today of relevance. And what you can see here, this is just a global map of Ciguatera fish poisoning. So that's the poisoning or toxidrome associated with ciguatoxin. And you can see here, based on the red dots, where known poisoning events have been shown to occur. And you can see it more or less occurs within the equatorial or temperate regions. And the main reason for this is because of their association, the algae's association with coral reefs. Ciguatera poisoning, one of the most significant forms of human food poisoning, has been attributed to roughly 50,000 cases worldwide per year with known mortality events. And like I've already stated, more associated with tropical, subtropical reef, and more specifically, the reef fish that tend to live on those reefs. And on the right, you can see just a bulletin that was put together by the Department of Health in Florida with respect to public awareness regarding Ciguatera poisoning. As far as incidence of Ciguatera, and on the left are countries or regions, several in the South Pacific Islands where you see kind of the highest incident per 100,000 people. But I've outlined in the red box where in North America, we also have to be vigilant in terms of keeping this on our differential, depending on the presentation of various patients that might be coming in. Whether you live in the US Virgin Islands or in the Southeast, including Florida. But also keep in mind, people are fishing in those regions and bringing sometimes their fish back to wherever they might live, whether it's the Midwest or anywhere else. Some of the most common fish associated with Ciguatera include the grouper, the barracuda, and the snapper. And these tend to be at the top of the food chain within those coral reefs. And as such, through biomagnification of this lipophilic toxin, they're accumulating in these fish, and then they tend to be our main game fish when we're fishing. And as such, we tend to eat these and then be exposed ourselves. The major symptoms associated with Ciguatera toxin, as far as early and late, early you might see more of a gastrointestinal symptoms. They come in, they're vomiting, they have diarrhea, and these symptoms tend to come on within the first several hours after ingestion. But there's also been documented cases of cardiovascular effects, including but not limited to bradycardia, heart block to varying degrees, as well as hypotension. Some of the late symptoms include more neurological, and these tend to be a little bit more delayed in terms of their presentation. Several hours to several days later, they might experience numbness, tingling in the extremities, and headache, vertigo, weakness, and some interesting phenomena such as temperature reversal, as well as this really interesting one that I don't think there's much of an explanation, but people complaining of tooth looseness. There has been associated mortalities associated with Ciguatera, and then there's a phenomenon called symptom reoccurrence. You can see here, this also occurs at a delayed time point. It can be several days to several weeks, and even there's some anecdotal evidence of months later, where some food items associated after an initial intoxication event, some food items that someone might eat, they tend to have those similar symptoms, especially the neurological ones, all over again. And they include not only those foods, but also physical activity. And although there isn't great data, it's thought to be due to remobilization of those lipophilic toxins and or possibly an immunological sensitization at a delayed time point. The ciguatoxins themselves, there's various different congeners, upwards of 40 different kind of toxin subtypes with different variability associated with the toxins. But there are at least two different backbone structures, and these are termed the Pacific ciguatoxin and the Caribbean ciguatoxin, as shown by a P and the C. And what's interesting is that even though the toxin structures themselves are very similar and they have the same mechanism, those subtle differences can actually lead to a different set of symptoms that those people might be experiencing depending on the toxin subgroup that they've been exposed to. For example, in the Pacific, there tends to be more of a predominance of the neurological symptoms. In the Caribbean, there tends to be more of a predominance of the GI symptoms. And then in the Indian Ocean, there's an additional set of symptoms that tend to have more hallucinogenic responses. Now, the mechanism of ciguatoxin is that these are activating voltage-gated sodium channels by binding to those channels and causing a persistent depolarization of those neurons. And we're going to talk about brevetoxin later, so I thought I'd put it up that these have a similar mechanism of action. On the left, you can see just a dose response curve. This is a competitive binding assay where you increase doses along the x-axis of the Ciguatera toxin. You're having reduced or displacement of another known ligand towards the voltage-gated sodium channels. And then here's just Pacific ciguatoxin plotted against Caribbean ciguatoxin. And you can see that Pacific is slightly more toxic as it shifted to the left. Here's kind of a cartoon version of the binding on the right side where the ciguatoxin actually embeds itself inside the voltage-gated sodium channel pore, and it has specific binding sites that have been elucidated through mutagenesis of the sodium channels as well as the toxin structures themselves, but that's something to be talked about another day. Specific treatment, if you have a suspicion for somebody, you have the history for someone who's been exposed to this, generally it's supportive care, and you're going to find that's a common theme amongst all of these toxin groups, which I'll be pointing out. But in addition to supportive care, EKG monitoring, especially for the intervals, QRS and QT, when you consider the mechanism on the voltage-gated sodium channels, is going to be important. And you can consider oral charcoal if it's been a recent exposure, and this was a recommendation that I'm not sure. Some of these recommendations you'll see are fairly outdated and really aren't backed up by kind of controlled trials, but nonetheless, there is consider at least activated charcoal. And then there's some debate as well, at least in the literature, whether you should use IV mannitol or not. And the thought there is that it may be helpful to reduce acute and subacute neurological symptoms. But consider that, but it certainly isn't a slam dunk in terms of its provision. And this is just a shameless slide capture out of Roche Review. And you can see here I've covered really many of these things, and this is what's been a lot of just emergency medicine physicians are going to be aware of when it comes to ciguatoxin and ciguatera itself. So with that, we'll move on to the next group, and this is saxitoxin. Here's kind of the global map. You can see it can be found globally, pretty much everywhere. And it's probably the most widely dispersed and maybe even widely recognized algal toxin, and it causes paralytic shellfish poisoning. And as you can see from the name, it also gives you some insight in terms of the symptoms. PSP, or paralytic shellfish poisoning caused by saxitoxin, can be produced by both dinoflagellates as well as cyanobacteria. And this is one that at least the US military and CDC are aware of in terms of potential bioterrorism abuses. And at least the ones that I'm familiar with are by taking large amounts of saxitoxin and putting it into freshwater water supplies. Now, it's caused, at least from kind of an environmental aspect, it's caused by the consumption of contaminated seafood, shellfish and fish. It's not destroyed by cooking or freezing, so it's not labile, and it's certainly lethal. Down at the bottom, I've shown kind of two trophic transfer pathways. On the top is more so associated with humpback whales, and this was off the east coast of the US, where several whales were washing up on shore in the late 80s. And you can see the bioaccumulation through some crustaceans, including krill, into the mackerel and then into the whales. But more commonly for humans, the human exposure routes are going to be through contaminated shellfish, and then more recently identified are pufferfish. The symptoms tend to be more rapid onset than some of the other toxin groups, within minutes to hours, depending on, I think, the concentration in the shellfish or the fish that you've eaten, as well as the total dose. And you tend to start to see very early neurological effects that include numbness of the lips and the fingertips, loss of coordination, drowsiness, nausea, paralysis, and then this feeling of floating that's been described by people that have been exposed. High doses can cause muscular paralysis, respiratory issues, choking, and then death as well, that can occur as soon as 2 hours after an ingestion. And the lethal dose for saxitoxin, at least in adults, is around 400 micrograms. And this is just out of Alaska. This is just a survey of patients in terms of their symptoms. And as you can see, these are very similar to what I've just talked about, primarily neurological as well as gastrointestinal. The toxins themselves, I'm not really going to go into these, but again, there's dozens of different toxin congeners or variants, all with different R groups on the various carbons that can lead to vast changes in the toxicity of these toxins, as well as the water solubility. The basic mechanism is through blockage of voltage-gated sodium channels, and this is by outer surface blockage at the neuronal membranes. And on the left, you can see percent inhibition on the x-axis or on the y-axis. On the x-axis, the concentration going from low to high, and you can see percent inhibition of voltage-gated sodium channels. And on the right, you can see just a cartoon version of how saxitoxin, and for that matter, and even tetrodotoxin, can inhibit these voltage-gated sodium channels. The treatment within the literature, it suggests gastric lavage, but I would be very cautious with gastric lavage as in terms of a recommendation. Maybe not very well supported in the literature, but nonetheless, it is a recommendation that's out there. Supportive therapy is, again, your number one recommendation, particularly for airway protection and ventilation for people that have ingested a large dose. And again, cardiac monitoring by EKG and monitoring those intervals. And then something that's had been worked on in the 90s, and I haven't seen much since, and there was a significant amount of studies done in both cellular as well as animal studies, and that's the use of 4-aminopyridine, which is a potassium channel blocker that had shown what appeared to be great promise in terms of reducing its toxicity. But I do want to state, and I've underlined it, that there's been no human studies here, and as such, it at this time should not be a recommendation for any patients that have been exposed. Moving on to our next toxin group, this would be microcystins. And you can, these are the prokaryotic phytoplankton or algae, and they're found everywhere. And the one thing I do want to state, in contrast to the other two that we talked about that were more coastal and marine systems, that microcystis and in turn microcystin can be found in freshwater as well as coastal systems, and can be found all across the world. Very close to home for me, Lake Erie. You can see here the Western Basin. This is some satellite imagery of the algae themselves, and this is an image that was given out of California, the Mbari Monterey Bay Institute. And you can see here, based on the light green on the western side of Lake Erie, and these are dense populations of microcystis, and in turn, through sampling by boat and such, are all producing microcystin. Now, exposure, as you can, I'm sure, guess, can be not only through recreational exposure. This is a guy on a jet ski in an Ohio lake, not Lake Erie, but it can also be found in freshwater or water supplies that is coming to our homes. And interestingly, you can find it in nutrient supplements. This is an industry that tends not to be very well monitored by the FDA. And through a pretty neat study taking blue-green algae nutraceuticals, we're finding pretty significant doses of microcystin in there that can have several effects, of which I'll describe shortly. In 2014, though, there was a state of emergency that was started by the governor in Ohio at the time, when over 100 people were showing up into the hospitals feeling ill. And at that point, the governor enacted the state of emergency and were told not to use the water in their house. And the World Health Organization limit in drinking water is 1 microgram per liter. And at that time, in the Finnish drinking water, there was at least 2.5 micrograms per liter during this episode several years ago. So the symptoms when people are exposed to significant doses of microcystin initially include some aspect of visual disturbances, but then mostly gastrointestinal, nausea, vomiting, diarrhea, abdominal pain, with some muscle weakness. But through a really unfortunate incident back in the late 80s, there was an episode in Brazil where a dialysis clinic was unfortunately using water that was not appropriately tested that contained significant amounts of microcystin. And at that time, 131 dialysis patients were exposed to microcystin, very high doses as it was being concentrated into their bloodstreams. And many of these people, in fact, died. Through subsequent animal studies, what we're starting to see now too is that there is significant liver toxicity and an association with non-alcoholic fatty acid liver disease that has been shown not only in some of those humans post after they had passed away, but also in some animal, both mouse and rat studies, and then through exposed sea otters, we're seeing a lot of this hepatotoxicity. And you can see some slides there, and these were taken out of some animal studies. I believe these were mouse that showed the more of the non-alcoholic fatty liver disease associated with microcystin on these H&E biopsy slides. Microcystin also tends to accumulate in the gonads, so there's been several in vitro studies looking at ovotoxicity. And then more recently, there's been some literature showing coagulopathy and thrombocytopenia, and this has been shown both in dogs that are drinking from freshwater ponds and such that are being exposed to significant levels of microcystin, as well as the sea otters that were discussed above that are experiencing thrombocytopenia and coagulopathy. This is a really interesting study, not necessarily cause and effect, but definitely correlation. This was a kind of a heat map of the US. On the top, you're seeing blooms or the presence of microcystin and the algae that produces microcystin across the US, so bloom coverage. And on the bottom, you're seeing basically the incidence of non-alcoholic liver disease. And through this study that came out of Ohio State in 2015, they correlated the risk of non-alcoholic liver disease, and it's increased by 0.3% with each 1% increase in bloom coverage. So something definitely to consider. The microsystems themselves, there's almost probably more than 100 by now. These are more of amino acid type structures that make up this toxin subgroup with several different derivations or congeners depending on the side groups. But the mechanism of action tends to be fairly conserved, and for microcystin, it's inhibition of serine threonine protein phosphatases, which is also similar to the diuretic shellfish poison toxins that I'll speak with you shortly about too. On the left, you can see protein phosphatase activity graph, and the activity is being inhibited by increasing doses of microcystin on the x-axis. And on the right side, you can see just a general schematic for how these microcystin toxins are affecting cellular metabolism, and it's postulated that they're producing significant amounts of reactive oxygen species, which results in cytotoxicity. And then through some other mechanisms, it's leading to DNA adducts, which is reducing the ability for repair. And then detoxification tends to be through more of the phase 2 conjugation with glutathione. And we can also see that through some of the toxin structures that are being found in some of the, not only humans, but also some of the animal models that the studies have been performed on. So treatment of someone who is exposed to microcystin toxins include, again, supportive treatment, especially controlling the GI symptoms. And then there is a recommendation for NAC, which I think we're all familiar with, and it showed pretty significant promise in vitro as well as in dogs. I don't think there's any human studies at this time, but I think through, if you're carefully weighing the pros and the cons of someone who's coming in with this type of a toxindrome, I think it should be a serious consideration. Now, less serious things to consider, but have more been shown in animal studies, is that of giving cholestyramine, which is speculated to affect enterohepatic circulation. That has been shown to have some promise in dogs, but there's no human studies. And then there's some other secondary metabolites, something called silibinin, which is a milkweed product, which has shown some promise in dogs, but I don't think that's ready for prime time yet. And then I just recently learned that through dogs that were exposed down in a Florida lake reservoir, they were exposed to very, very high doses, several of which of these dogs died. Plasmapheresis had been used, which they were speculating had helped reduce mortality of the few dogs that did happen to survive. Moving on to another toxin class called azospiracid. And this is one that is very near and dear to my own heart. This is one that started to be identified in Western Europe in the late 90s, and I got involved very early on with this toxin group, and it's been also shown to have um, human health effects to several campers that were unfortunately exposed in the Puget Sound region just outside of Seattle. And so these are the known human health poisoning events associated with azospiracid. The toxin structure itself, there's at least 30 or 40 different toxin congeners. It's produced by a particular type of eukaryotic algae called azodinium, and the human exposure route is by shellfish. Early on, there's acute gastrointestinal symptoms, vomiting, diarrhea. This can occur within minutes to hours after exposure. And then symptoms typically resolve in several days with no other known side effects, at least to date. And on the right, you can see that this is a colleague of mine out of Japan that published some mouse data, and this is looking at small intestinal microvilli. And on the left, you can see the azospiracid exposed mice to the microvilli, and on the right would be the control that were not exposed. And this is through scanning electron microscopy. And some of her same data out of Japan had shown that there was increased intestinal hyperplasia, as well as an increase in intestinal cancer in the mice that were exposed over long periods of time. The mechanism, the only known mechanism thus far for azospiracid is work that's been done out of my lab, where we identified this as an open state potassium channel a blocker. But I do want to point out that that mechanism that's been identified so far doesn't necessarily correspond yet to some of the intestinal effects that we tend to see in humans that are exposed. And to date, there's been no known cardiac or neurological symptoms associated with the few humans that have been exposed to azospiracid. The treatment is predominantly supportive care then for the GI symptoms, but also considering the mechanism, I would very much consider cardiac monitoring and EKG also to look at the intervals. The next toxin group that I'll discuss is brevetoxin. Global distribution is predominantly in the Southeast US, but there have also been some known exposure events in New Zealand. And this toxin group, brevetoxin, is produced by a species called Carenia brevis. And through kind of newspaper reports and such, it's coined the term red tides, which is really kind of interesting because it's neither red nor is it directly associated with tides. But nonetheless, if you live in Florida or if you're familiar with people who go to Florida, this is what this particular toxin class is associated with, red tides. Now, the exposure to this toxin class is both through ingestion as well as through aerosolic toxin exposure. So for the ingestion exposure, human health of symptoms tend to occur within minutes to hours, and it again tends to be more gastrointestinal, nausea, vomiting, diarrhea. But there are known neurological effects, paresthesias, ataxia, limb paralysis, slurred speech, headache, meiosis. And so I think it's important here to recognize that a lot of these might be very similar to stroke-like symptoms. And again, where the getting the HPI of someone's potential exposures is so important. And if I had a patient like this, quite frankly, I'd be very likely to activate a stroke pager until it can to ensure that they're not having an acute stroke at that time. Respiratory symptoms tend to occur within seconds. So this might be someone walking on the beach. They get the ongoing winds are going onto the beach. And as such, through just wave action, these algae tend to be fairly fragile. And there's known aerosolic doses of the brevitoxin that can come on shore if the winds and if the wave action is appropriate. The symptoms tend to be more respiratory. You get a cough, you get throat irritation, chest tightness, wheezing, dyspnea, as well as pruritus and headache, eye irritation. And the people that tend to be most susceptible to this are not only the extremes of age, children, as well as people that are older, but also those with known lung diseases, asthma, and COPD. And through some of the studies that have been done, even in healthy adults, there's been shown to be significant decreases in the forced expiratory volumes as well as peak flows. And a lot of these studies have been almost occupational exposure to lifeguards. And in many cases, it's lifeguards that are being recruited as well as beachgoers into many of these studies. The toxins themselves also very diverse, but they do have a general backbone. And then there's the A and B types, but again, they have the same mechanisms of action, which are activation of voltage-gated sodium channels. And this is similar to the SIGWT toxins that I've talked about previously. A lot of the data initially came out of Bill Catterall's lab up in Seattle, and through mutagenesis of the voltage-gated sodium channels, it's been shown that the specific binding points tend to be within domains 1 and 4, more specifically segments 5 and 6 of the transmembrane portions of the voltage-gated sodium channel, where the toxin appears to embed itself directly into the channel. On the left, you can see here, this is just a competitive binding assay again, where brevitoxin is displacing known ligands towards the voltage-gated sodium channel at increasing doses. Treatment of brevitoxin is, you have to keep in mind the exposure mechanism, whether it's through ingestion or whether it's airborne exposure. Supportive care, again, is going to be your #1 goal in many of these cases. And respiratory monitoring, also very important when it comes to both of the exposure mechanisms. Activated charcoal has been a suggestion if exposure by ingestion has been within 4 hours. But for those that are being exposed through aerosolic exposure, you should consider, especially in the face of asthma or COPD exacerbation, you should be considering your airway adjuvants in terms of smooth muscle relaxation and albuterol and ipratropium. Cardiac monitoring is also going to be important, especially when you consider the mechanism. And as I've already stated, if someone's having neurological symptoms, you should be ruling out a stroke just as quickly as figuring out whether you think this is a brevitoxin exposure. I think there's been some literature suggestion of use of IV mannitol, which I think was really, quite frankly, just been stolen from the Ciguatera literature, but I would only consider this. I definitely don't think this is a slam dunk in terms of its provision. And then some animal studies are starting to show some promise towards a natural antagonist called Brevinol. And this has been been developed out of a lab at the University of Georgia, I believe, but no human studies have been performed with this to date. So moving on to okadaic acid, okadaic acid belongs to a group in the diuretic shellfish poisoning. Again, the symptoms you can very quickly ascertain based on just the name itself. And you can see here the global distribution is very broad, with quite a distribution that is focused especially in Western Europe and Scandinavia. The source, they're produced by different types of algae, and then the toxins themselves. There's two different subgroups, but the toxins are very similar: okadaic acid and dinophysis toxins. And then the exposure route is predominantly through shellfish, so the consumption of contaminated shellfish. Symptoms tend to be within minutes to hours. And then if you're not having any of the symptoms within 12 or so hours after the exposure, you're not likely to exhibit any of the symptoms, which tends to be diarrhea, nausea, vomiting, abdominal pain. And then severe symptoms, there is a concern of tumorigenesis for these particular toxins, but that's been something that is more postulated, not very well supported, at least in human exposures. The mechanism of action of the diuretic shellfish poison toxins, dinophysis and okadaic acid, is also the same as microcystin, despite the very diverse toxin structures. And that's through the inhibition of the serine threonine protein phosphatases and protein phosphatase 1, 2A, and 5. And what this does is through inhibition of those protein phosphatases, you're losing the control for sodium channel secretion in intestinal cells, which is resulting in vast volumes of watery diarrhea. And on the left, you can see here, this is just enzyme activity on the y-axis, and this is taken out of one of our papers, where we looked at various toxins within this group, doing structure activity relationship studies. And you can see here there's certainly some diversity in terms of the EC50s associated with inhibition of these protein phosphatases. And by inhibition of protein phosphatases, what you're essentially doing is you're kind of disrupting the balance between the kinases and the phosphatases, leading to overrepresentation of the active enzyme in its phosphorylated state, which in turn then results in sodium and water losses from the intestinal cells. Treatment of these particular toxins, again, supportive care, IV fluids, antiemetics, electrolyte replacements, and I think, as can be said for all of them, reducing additional exposures that can lead to potential tumorigenicity. So moving on to the last one that I'm going to talk about, and this is domoic acid. Certainly not its last, but not the least interesting. This can be found also globally. You can see it in Australia, New Zealand, Western Europe, and across North and even South America is where it's been found and associated with human events. Now, it's produced by a eukaryote, which is, in this case, a diatom, and which is basically a small silica shell, as you can see in the electron microscope image on the left side. And then you can see here just an image of North America coastlines and known incidences of where domoic acid can be found. And what's interesting is that maybe whether it's folklore or truth, I'm not really sure, but it's been postulated that Alfred Hitchcock, when he was on the West Coast, close to San Francisco, seeing the erratic behavior of birds in that region, which may have been exposed to domoic acid, causing some of that erratic behavior, was the inspiration for his famous movie called The Birds. Domoic acid itself initially and very quickly identified in the late 80s, shortly after people in Prince Edward Island, Canada, were being exposed. More than 100 people, and 4 people subsequently died. And that was through the domoic acid accumulating in blue mussels that were then being exposed to those folks that were eating the shellfish. But more commonly, what we see, at least in North America, we see marine mammal events where marine mammals, especially California sea lions, are feasting on shellfish as well as other organisms lower in the food chain and exposing themselves to very high doses of domoic acid and resulting in their mortality events as well, where you might have hundreds of these animals that are washing up on the shore. And as you can imagine, the news headlines associated with that. The symptoms associated with domoic acid include very early on GI symptoms as well, very common for many of these toxidromes. But then the delayed symptoms are quite unique. You can have headaches, dizziness, hallucinations, confusion, short-term memory loss, respiratory difficulties, tremors, seizures, coma, and death. And the activities of these toxins can actually be seen when you look at the actual toxin structures themselves. On the left, you have a domoic acid molecule, and you can see the two carboxylic acid groups there. And you can see kynic acid and glutamic acid, which are known neural excitatory neurotransmitters within our cells. And so the mechanism of action of domoic acid is based on the chemical similarities with the similar to glutamate, and that it essentially mimics the excitatory neurotransmitter glutamate, more particularly to the ionotrophic glutamate receptors. And through structure activity relationships, it's been shown, or studies, sorry, it's been shown that the kynic acid receptor is more sensitive to domoic acid than is the AMPA receptor, which is more sensitive than the NMDA receptor. Actually, and what you can see here, just in terms of this mimicking behavior, on the top of that little synapse, you can see the presynaptic neuron, and then on the bottom, the postsynaptic neuron. And through those mimicking actions of domoic acid, you have an increase in the postsynaptic intracellular calcium, which leads to several downstream secondary signaling side effects, as well as apoptosis within the neurons themselves. When you look at where these particular channels are being found within animals as well as humans, they tend to be located more in the temporal lobe regions, with a specific increase in the numbers in the hippocampal, which likely explains the short-term memory loss associated with this toxin group. Known seizure effects, and this is one of the more interesting things that you can see, and this is through and been corroborated through EEG, as you can see up there at the top, looking at seizure activity, but also in these sea lions, you can see the this is a typical seizure, and you can see the posturing associated with the episotonous posturing during this particular seizure event after this unfortunate sea lion was exposed. And then my colleague in Seattle also has done a lot of mouse studies, and what you can see there is some hind limb shaking, which appears to be unique to the domoic acid class as well. Treatment of domoic acid poisoning is, again, supportive care. You should also consider seizure precautions because of their known incidence for causing seizures. And the recommendations for seizure abatement is benzodiazepines, which are first line, followed by propofol and barbiturates. And then one thing that I found just by looking more for this talk is that, at least out of the California Poison Control Center, there's a recommendation on one of their brochures as far as the provision of valproic acid and vitamin B6. But I wasn't able to find any primary literature to support this. And then also in some of the sea lion literature, the provision of alpha lipoic acid has shown some promise in sea lions that are experiencing seizures. And so with that, we've really gone through all of the different toxin groups that I think are important from an emergency medicine point of view and a poison control center point of view, not only here in North America, but also in, I think, globally, because this is not just a North American issue. These are global issues. And what I've done on this slide is really just, I'm not going to go through it, but just kind of outlined each one of the toxin classes or toxindromes that I've gone through today. And on the far right, kind of put unique symptoms associated with each of those particular poisoning events. And so I think this is important for us to recognize, whether it's for your boards or whether it's for a patient you might see that comes into the EDs, or you might be getting calls on at a poison control center, to be able to recognize the potential toxidrome, or maybe I can coin the term algodrome events. And that although there is some geotemporal variability, I think with kind of the global landscape of and travel of people, and I've already mentioned for example, you might be down in the Caribbean, you might be caught a huge barracuda, but you don't actually eat it there, you bring it home for the whole family. I think it's, again, the importance of it, the HPI and recent travel that can help tease apart what the actual event is, or what the poisoning toxidrome actually is, without the analytical resources to be able to identify these toxins in their blood or their urine or otherwise. And then I've also alluded to the importance too for the potential large scale events. And again, the US military is aware of this, and they're also researching, and saxitoxin's the one that I'm aware of, and that's kind of the bioterrorism event of where someone might take a large amount of saxitoxin, which is highly potent, and put it in a freshwater supply, potentially exposing thousands, if not tens of thousands of people. And so with that, I'd like to just say thank you to the people that helped me along the way in terms of provision of some of these slides and videos, as well as Nick and all of ACMT for inviting me here, as well as Andy King, which is local, and pass my name forward. And so with that, thank you for your attention. I can be reached at this email here if you have any questions that you don't wish to ask here today. And then after you leave, maybe one of the most interesting things I do with some of my students is pull up this video, and this was in Oregon back in the 70s, I think, and this was a whale that washed up on shore that was likely full of algal toxins, and how the Corps of Engineers dealt with that. So with that, any questions I'd be happy to entertain, both here live or alternatively later by email.
Dr. Twiner, that was excellent. Thank you very much for taking the time to come talk to us, and really found that to be a fantastic deep dive into some of the things that that we talk a lot about in toxicology. We do have one question that came up already. Early on the talk, George Wang wanted to ask, what is the cause of death in cases of ciguatera associated death?
Yeah, great question. The general cause of death for that particular toxin class is believed to be through more of a respiratory issue and a neurological issue. So it's likely through the sodium channel activation leading to respiratory paralysis is the postulated mechanism by which people exposed to enough of the toxin are experiencing death.
See, if you do have a question, if you could use the raise your hand function that brings you up, it makes it easier for me to unmute you. Try to find. Just 1 second here. Attendee, there it is. Well, in the meanwhile, I had a couple of questions. Are you familiar with any cases of microcystin toxicity not associated with dialysis in humans? And also, you pointed out that potentially this could be a cause of an undifferentiated liver failure or liver toxicity. Is there any biosample testing that is available?
Yeah, two really great questions. The short answer of microcystin exposures, I mean, the Toledo event was certainly one where the hundreds of people were showing up complaining of those symptoms. But as far as mortality, no, there's no documented cases of microcystin causing mortality in humans. But there's hundreds of cases of microcystin that is in like farm ponds and things like that. And then you have livestock as well as dogs and other terrestrial animals that are being exposed just by drinking water from those ponds. But no, other than the dialysis event, there's no other that I know of human deaths associated with microcystin. As far as biosamples, um, so when these events are known to occur, at least what tends to happen is it's more of a state regulation and guidance, usually the Department of Health, that is going to get involved and help coordinate sample testing. And this is, I think, something that could use some refinement in terms of the toxin classes. But microcystin itself, if we're talking about just microcystin, I mean, there's animal studies that are showing microcystin in serum as well as urine, and then even hair is being shown to be able to kind of provide a snapshot of exposure events, both acute as well as maybe even subacute. So I think some coordination of efforts is going to be done at the Department of Health level to get those samples to the right agencies, whether it's themselves or whether it's going to the FDA or the CDC to get the appropriate analyses done.
Thank you. Michael Yee, unmuted. Are you there? Michael Yee? Remute you and we'll go on to the next person. Eduardo Rosa.
Great talk. I have two questions for you. Do you know if domoic acid actually affects the Kv1.1 channel on the paranode of myelin?
I'll be honest, I don't have a specific answer for that. I know there's a lot of groups that are certainly, studying the actual subtype effects of domoic acid, but I don't offhand know the answer right now.
Yeah. Have you seen any demyelinating effects with domoic acid?
So I personally also have not seen that, of the toxins here that were presented. That's probably, I mean, domoic acid, unfortunately, is one of the ones I've not worked on myself. And I also, unfortunately, don't know the actual answer whether they're known demyelinators or not.
Yeah. And my last one is, what about Fisteria? I mean, have you done any work with Fisteria?
Yeah, so Fisteria was something that was came out of like North Carolina, and was a really big deal in the late 90s and 2000s. What's speculated now, kind of in retrospect, after all of the quote unquote Fisteria hysteria, is that Fisteria was actually likely attributed to a different toxin class called carlodinium toxin. And the Fisteria alga itself was a heterotroph, so it was feeding on other types of algae, which was likely not producing the toxin itself. And so I'm not sure if there's true closure of the Fisteria world itself. Other than that, I think the most of the people I've spoke to believe that it's a different toxin class that I didn't speak about today called carlodinium toxin, which, if I'm not mistaken, causes calcium channel effects. But a good colleague of mine, Jonathan Deeds, who's now at the FDA in Maryland, has kind of headed up that work. But I don't think there's really any additional research going on in that field at this time.
Thank you.
You're welcome.
Dr. Kazi.
Yes, hi, hello, how are you? Can you hear me?
Yep, you're on.
Great, thanks. I have a question. I took care of a patient last year around this time of year. He had ciguatera poisoning, quite severe. He was in the island of Mystique in the Caribbean. He had assured me that this was the first case that was seen from eating barracuda on that island, although barracuda was a commonly eaten fish on that island. A few months later, I was in St. Lucia, which is near Mystique Island. And I asked one of the restaurateurs there about this question, and they serve barracuda quite regularly in that area. So, they were doubting that barracuda would cause this, although, as you have mentioned, and I had always believed, barracuda was a common culprit, or was one of the culprit fish in ciguatera poisoning. Can you comment on that? How come these people were like so completely, unfamiliar with this link between barracuda and ciguatera? And my second quick question is, where do you see the research needs for ciguatera poisoning? If we are going to apply for some funding, what topics you think are worthwhile researching? What questions need to be answered?
They're really great questions. So one of the interesting things, at least in the Caribbean and associated with ciguatera poisoning, is that many of the islands do recognize that it's an issue, but also many of the locals have also identified, and this is probably through trial and error, quite frankly, which reefs are safe and which reefs are not safe. And barracuda, although they can be somewhat migratory, tend to be more so floating around particular reefs and tend, at least in the past, tend not to go to a nearby reef. So they tend to be very local. And what was always interesting, again, was that the locals would know, okay, we can go fishing on this reef, but not this one. But I think the hazard with that is that with times changing, there's known reefs that are dying off, whether it's through water warming or whether it's through acidification of the waters. You're going to have known, as these reefs are dying off, you're going to have a change in the ecology and the biology associated with those islands. And so you might actually have barracuda that are then moving to other reefs, and local folklore might have to change that too. But certainly barracuda are associated with ciguatera toxins, but it tends to, at least historically, be more associated with specific reefs because of the feeding behavior of those barracuda. And I have several colleagues that have studied barracuda themselves and showing very high concentrations of barracuda. As far as future funding, I think there is significant efforts here, at least on the biological side and the ecological side. So, I would be more than happy to put you in touch with some of my colleagues, both at University of Southern Alabama as well as Woods Hole Oceanographic Institute, because they're doing a lot of work now as far as looking at reef ecology, looking at the toxins themselves as far as biomagnification and what specific species. And furthermore, and I think what's really needed are rapid identification mechanisms. So whether it's like a pregnancy test, like a lateral flow device where you can take a ground up fish and put it on the device and it tells you whether it's safe or not. I think these are all things that are really needed as far as protecting human health and identifying rapid detection methods. But I think what's also needed, and this is where I see myself getting involved in this too, is linkages towards human health and human treatments. And so I think maybe one of the best things that could be done that I would recommend is hooking up with some of those other basic scientists and, where you could approach this from a human health side effect. and they could maybe approach it from more of the ecological side.
Thank you. We've submitted 2 grants in this area. Unfortunately, they were not funded, but one was to look at the prevalence of ciguatera-like symptoms. 3 or 4 islands in the Caribbean, including St. Lucia, with the Ministry of Health. And the second thing we wanted to do was link it up with some sort of telemedicine, clinical care, as well as education network through maybe a database, sentinel sites in the Caribbean to surveil for these illnesses. But unfortunately, we were not successful. We hope to try again. I'll shoot you an email to follow up.
Please do.
Thank you.
Thank you.
Wonderful. Thank you guys very much. Thank you again to Dr. Twiner, and we'll see you at the next ACMT Grand Rounds.
Okay. Thank you very much, Nick, and everybody else for listening.